A very big hello to my wonderful PU2 champions. I hope all my dear children are doing absolutely great. So this is something that you asked from us, and that is model question paper solution. And if at all by any chance you guys have any plans to not solve model question paper before going into the examination hall, then I'll say it's a very wrong decision, my dear children. Please make sure you're solving it. There is definitely a purpose why government releases model question papers, 3 model question papers every single year, right? So that you can understand what is the question pattern, what could be the difficulty level, what are the concepts which are repeated, everything. And I'll tell you what I got to understand while analyzing this model question paper, that is model question paper 1. So over here, the 1 marker, the 3 marker and the 5 marker, they are totally fine, they're good to go. Comparatively, repeated concepts out there. But if we talk about the 2 marker questions, here you find new things, some things which are often not repeated so much. So that's the newness in this particular paper, which is why I'll recommend you to watch it fully. And if you think that this video is helping you, what do you have to do? You have to click on the subscribe button. Wishing you all the best, my dear children. Let's go ahead with the first question. Statement 1 says, pollination does not guarantee the transfer of right type of pollen. Pollination, my dear children, suppose there's a flower over here which has the male and the female part. Now from some other flower, pollen grains are landing over here. Now can you guarantee that this is the right type of pollen which is coming? I do not know the species of that particular flower from where the pollen is coming. So it can be right type, it may not be right type, but pollination is definitely happening. Pollen grains are landing on the stigma for sure. So can I say that pollination can happen, but it does not guarantee that the right type of pollen is there? Yes. So pollination does not guarantee the transfer of right type of pollen. This statement which is there, it is absolutely correct. Now going ahead with the statement 2. Compatible pollen-pistil interaction guarantee double fertilization. What do you mean by compatible pollen-pistil interaction? So suppose over here you have the stigma of a plant, stigma and the style, everything, and your pollen grain just landed on it. Now if it is of the same species, everything is same, my dear children, what will happen? Basically you say that this is compatible. When the chemical interaction is happening over here, it's totally fine. We say that this is compatible, which is why the pollen grain which is there, it will form a pollen tube. It forms a pollen tube and transfers pollen tube and transfers what? Gametes. How many gametes? There will be 2 gametes per pollen tube. Okay. This is what you mean by compatibility. Now do you think that it will guarantee double fertilization? That is a whole different thing. What do you mean by double fertilization? Means 2 times fertilization is happening. Where, ma'am? In the ovule. What is there in the ovule, ma'am? In the ovule, my dear children, you have the embryo sac. And the embryo sac, it has different types of cells. This cell in the middle that you see is known as the egg cell, the female gamete. So one male gamete comes and fuses with the egg cell. You get the zygote as the product. This is one fertilization. Okay, this is the first fertilization. Now one more fertilization happens. Why? Because you have one more gamete, right? It also has to fuse with something. So basically this thing, the bigger green cell that you see over here, it's a big cell which has 2 nuclei. It has 2 nuclei. This is the nucleus. Now the second male gamete comes and fuses with the 2 polar nuclei. You call them polar nuclei. And together now it will be what? It will be forming something which is known as the endosperm. So you got zygote from the first fertilization. From the second fertilization you got something which is known as endosperm, which is going to nourish the zygote so that the zygote can divide, form the embryo properly. So 2 times fertilization is happening. Now do you think just because there is compatible pollen-pistil interaction, all of this will happen? No, that is not a guarantee. Sometimes the pollen tube can grow until here and can stop. So basically this statement, which is second statement, compatible pollen-pistil interaction guarantees double fertilization, would be wrong. This is incorrect, but the other one is correct. So your answer for this question would be what? C. Statement 1 is correct, but statement 2 is what? Incorrect. Understood? I hope you understood. So there are multiple pollen grains which can land, multiple pollen tubes which are formed. Only one will be able to go around and do the double fertilization. You need to understand that. Okay? So if you have understood, just give me a quick like, and my dear children, share it with your friends also who are in need. Now the secondary oocyte formation from the primary oocyte occurs in which of the follicle? That is a question. So there is primary oocyte. Okay, there is primary oocyte and there is secondary oocyte also, which is basically formed when the primary oocyte is going to divide. So the primary oocyte which is there, it divides into two, one small cell and one big cell. The big cell is known as the secondary oocyte and the small cell which is there, it is known as the polar body. Okay, the polar body. You're talking about how you need to make the gametes. So basically the secondary oocyte which is there, it will later divide again. It will form what? It will again form some PU2 body which is known as the second polar body. And the major thing which is being formed is the ovum, which is the female gamete. But this is not that easy. It happens very, very slow. So primary oocyte, when it is being formed, it is usually covered up by some granulosa cells. Some cells which you call as granulosa cells. Now together you call this structure as the primary follicle. The primary follicle. Got it? Now later what happens? The primary oocyte remains like that only, but just above this primary follicle, there are some changes happening. There is layer of granulosa cells being added up. There is one more layer of granulosa cells being added up, and not just that, there will be a theca, a new layer which is known as a theca layer, which also gets coming in over here. Now you would not call this as a primary follicle, rather you will call this as a secondary follicle. Remember, over here within that primary oocyte is there. Within the secondary follicle, the primary oocyte is there. It's nothing else. Okay. Now what will happen, my dear children? Later on, we want to transform the primary oocyte to secondary oocyte. Where do you think this happens? So basically, now the secondary follicle which is there, it is going to transform into the next type of follicle which you call as the tertiary follicle. So over here, you just had one theca, right? Now the theca differentiates to inner theca interna and outer theca externa. So basically, you have two theca layers now, and not just that, you will be having a small fluid-filled cavity right over here, which you call as, you would be calling this as what? Antrum. You will be calling this as antrum. And now over here, the primary oocyte which is there, it will divide, and what will happen to this primary oocyte? Basically, when the primary oocyte divides, you get a small polar body, and also you get what? The secondary oocyte. So it is inside the tertiary follicle, my dear children, within the tertiary follicle where you have the secondary oocyte. That is the question. The secondary oocyte formation from the primary oocyte occurs in which of the follicle? It is the tertiary follicle. Okay? Always remember that. Cool? So in primary, you had primary oocyte, primary follicle, you had primary oocyte. In secondary follicle also, you had primary oocyte one step behind. Similarly, in tertiary follicle, you have one step behind, that is secondary oocyte. It's not tertiary oocyte, okay? Chalo. Next question, question number 3. Progenies with AB and O blood groups are not possible from which of the following mating parents? AB and O should not be coming. AB and O should not be coming. So you are given these options, you have to tell which is the correct option where AB and O are not going to be formed. Over here, do you think a cross between them will not give you AB? Array, from here you get B, here you get A, so AB is there anyways, so cut it off. Now you look at this particular thing. Here just B is written, that means it is BO. Again, my dear children, if you cross this and this, you get AB. So I cannot take this into consideration. Now if you take this one, again AB is there. Again, this is not under consideration. What I am left with? I am left with this, O and AB. Over here, if I cross, it is AO. That means A blood group is there. If I cross this with this, again BO, that means B blood group is there. Again this with this, AO, A blood group is there. Again this with this, BO, B blood group is there. So only A or B will be there, not A, B or not O also. So your answer for this particular question would be what? A. Okay, your answer for this question would be A, because you are being asked what is the, which mating parents will not give you A, B and O. Now going ahead with the fourth question out there, which says, another chemical name for the nitrogenous base thymine is, what do you call thymine as? So you know what? Initially we had RNA, and RNA basically was later known to be something which was not stable, and because of evolution we got DNA. So DNA had to be more stable because you are changing something which is not good to something that is better, right? So obviously you need to make changes in the internal structure also. So in the RNA you had 4 nucleotides or 4 base pairs. One is adenine, next one is guanine, third one is cytosine, and the fourth one is uracil. And it was found that uracil is something that is causing trouble, so why not add some group into uracil and change it to something that is more stable? So it was done, and on the uracil, my dear children, on the 5th carbon's position, a methyl group was added. So it is known as 5-methyl uracil, and together you actually call it by the name which is thymine. So it is 5-methyl uracil, do not forget it. Your answer for this question would be D. Okay? Chalo, next question. Question number 5. Select the correct match. Australian marsupials are over here and placental mammals are over here. So you know what usually happened is that basically there was a big field. Just imagine there was a big field and there was A group of organisms which are marsupials, and there is P group of organisms which is placental mammals. Now from this big group, some of them moved here, 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 some of them moved here. While they move to different locations, obviously they have to get habitual to the location that they are coming in, right? So obviously if at all some organism came from the marsupial category, from the M category, and some organism came from the placental category, both of them will have something in common because they are living in the common area, my dear children. Okay, they are living in the common area. So both of them showed adaptive radiation. They moved to different location, and now they have some similar character. When different organisms have similar character, what do you call it? Convergent evolution. They are converging to a point where the habitat is such like that the organisms have to have common features to get adapted. Okay, so now you are asked, spotted cuscus is from the marsupial side. Which is the other organism from the placental mammal side? So your options are flying squirrel, bobcat, anteater and lemur. What is your answer? So this is a question that usually was not being asked. So this time they have asked it, and you should know the table then. You should then in your NCERT textbook, in adaptive radiation, just after that you have a table of these organisms. You should be knowing that. But I'll tell you, it's super easy to remember. Okay, so over here spotted cuscus actually goes with lemur, and this is the way to remember. So this is a table given to you, and my dear children, if you see mole, marsupial mole is there. The name is similar. Okay, mole and marsupial mole is there. Anteater and you have numbat. Anteater and numbat. This is a different name, which is why you might find it difficult to remember. But anteater, numbat, somehow you can think it's same only. Mouse, marsupial mouse is easy. Lemur and spotted cuscus, this is what we just discussed. Now flying squirrel, flying phalanges, this is easy because both of them have flying thing. Bobcat and Tasmanian tiger cat. Cat is similar. Wolf and Tasmanian wolf, that is also similar. So what is different? Here anteater and numbat, and also what? Lemur and spotted cuscus. This is what you need to remember. And also remember, spotted cuscus comes in which category? Basically they are marsupials. Okay, they are marsupials. And over here comes the placental mammal. So remember that. Mammal, squirrel comes under mammal. Phalanger comes under what? Marsupials. Cool. So these are the things that you need to remember as a new thing for this academic year's board paper. Now question number 6. Which of these ancient men protected their body with hide and buried their dead? So in the evolution of man, you might have seen some organisms, basically they were very concerned about naked body. They used to cover their body, and also they had emotions. They started burying their dead, had rituals around it. So which one do you think is the first one to do that? Homo habilis, Dryopithecus, Neanderthal man, Homo erectus. Your answer for this question is Neanderthal man. Okay, it is Neanderthal man. Always remember. I'll tell you one thing. So from the starting point onwards, you had two types of organisms. One is Dryopithecus, another one is Ramapithecus. Rama sounds like a man's name, right? Rama. So basically, they were the organisms which were closer to human beings. Dryopithecus were more ape-like. Okay, they were more like monkeys, apes. But Ramapithecus was more man-like. Now, from this Ramapithecus, you got differentiated into different things. So from Ramapithecus, you got Australopithecus. From Australopithecus, you got the first human-like organism, Homo habilis, Habibi. So that is this, Homo habilis. Now, the organisms which were being formed, they started to stand erect. They started to have meat. So from Homo habilis, you got Homo erectus. Okay, and from Homo erectus, you had two things. One is Neanderthal man, who used to hide their body and bury their dead. And the second is the modern day Homo sapiens, which is us. Okay, so here is a chart which you can use for remembering things. I have put down everything. Like 15 million years ago, you have Dryopithecus and Ramapithecus. Then all of this information has been given to you. Make sure you read it. It is super important for you. And you should be also knowing the years. That is also important. Okay, because in the previous years, Match the Following question has also come from this. In the Brahmastra series, I am solving the previous year questions. You can watch it and make the best use of it. Okay, so for this, the answer is B, Neanderthal man. Fine. Now, human immunodeficiency virus replicates in B cells, NK cells. NK cells stands for natural killer cells. Helper T cells or PMNL. PMNL is what? Polymorphonuclear leukocytes. That means their nucleus, my dear children, have multiple morphology. They look different. Okay, they have multiple lobes. So PMNL is a short form, or basically you call this as neutrophils. It's another group of cells which are known as neutrophils. You know what they do? They are phagocytes. Basically, suppose there is a pathogen over here. They will take this pathogen inside and kill them. Okay, they are phagocytes. So, ma'am, macrophages, that is a separate category. Okay, macrophages is a separate category. They also have a similar function. But usually when something comes, these are the first ones to come. These cells are the first one to come in the line of defense. Okay, anyway, the question is human immunodeficiency virus. That means HIV virus replicates in what? So we all know HIV has one function, to make sure that the helper T cells are down. They are not there, because helper T cells, they help in providing immunity. They are like the mediators which tells the other cell that, hey, you have to do this, hey, you have to secrete this, and it comes in the middle. So if at all we destroy the mediator, don't you think our whole immune system goes down? So basically, HIV viruses would be replicating in helper T cells and also killing these cells, which makes our immunity go low. But if at all, you have been asked this question, what is the HIV factory? Which cell is the HIV factory? And over here you have one more option, that is macrophages. That is macrophages. Then your answer should be macrophages, because when HIV virus comes, they do not directly get into the helper T cells. Rather, first, they attack the macrophages, they make sure they are replicating, they are increasing in their number, and then they go around killing the helper T cells. They also replicate within them, they also kill them. So here macrophage option was not there, which is why we chose this. But if at all you have an option like macrophage, it is said that which is the HIV factory. Macrophage is actually the HIV factory. Now, which are the cells which are being targeted by HIV viruses? It is actually helper T cells, because this makes our immunity go down. Done? Okay. Next question, question number 8. The drug which is depressant and slow down the body function is? The drug which is depressant. So, in your NCERT, there is one depressant drug, and that is heroin. That is what? Heroin, my dear children. So, heroin is not given as an option over here. That means we need to know the other name of heroin. What is it? Is it crack? It is hashish or charas. So hashish and charas altogether are different things, because they come under a different category which is known as cannabinoids. Crack is the other name for cocaine. Crack or coke is the other name for cocaine. What are we left with? We are left with smack. So smack is like heroin, which is a depressant. SMAC is the other name for this, which is a depressant. So heroin, commonly known as SMAC, is chemically diacetylmorphin. So other name of heroin is diacetylmorphin. So from the morphin, you do acetylation in morphin, and that's how you get heroin, okay? Which is why it is known as diacetylmorphin, which is white, odorless, bitter crystalline compound. This is obtained by acetylation of morphin, which is extracted from the latex of poppy plant, which is Papaver somniferum. So poppy plant, if you talk about its actual name, it is Papaver somniferum, and over here the latex of it provides you with this particular drug, that is heroin. Generally taken by snorting or injection, heroin is the depressant and slows down the body function. Okay, cool. Now question number 9. Saccharomyces cerevisiae, commonly called as brewer's yeast, is used for fermenting malted what? Pulses, cereals, fruits or vegetables. Saccharomyces cerevisiae, commonly called as brewer's yeast, is used for fermenting malted. Malted means basically what do? You allow the things to germinate, okay, the seeds to germinate, and then you stop it. Then you do not provide all the resources for germinating. You just stop the whole process and you keep it like that. Why is it being done? Because, my dear children, whenever these seeds which are being used up, if at all germination has started up, what happens is that the starch which is there, the storehouse which is there, it is being broken down to simpler sugars. It is being already broken down to simpler sugars. Now it will be easy for us to digest it. Otherwise we have to start from the beginning. So which is why you used malted cereals. Okay, so over here Saccharomyces cerevisiae or yeast is used for fermenting malted cereals. Fine. Now these malted cereals, when they are fermented, it is even more easier for the yeast to break them down. Okay, and that's how you get the brewery, that is your wine and everything out there. Okay, so it is cereals, not pulses, not fruits and not vegetables as well. So brewer's yeast is used for fermenting malted cereals and also fruit juices. The fruits are not malted, okay, fruit juices. Now this is from the chapter Microbes in Human Welfare. Going ahead with the next question, that is question number 10. In the year 1963, the two enzymes responsible for restricting the growth of bacteriophage in E. coli were isolated. One of them cut the DNA and other one added to the group. So there are two teammates, okay, two enzymes in an E. coli. Suppose this is an E. coli. In the E. coli, you have two enzymes which is there to basically restrict the growth of any other organism within them. So suppose a bacteriophage came one day. Bacteriophage, you know what is it? It is a virus. And viruses basically, they transport their genetic material inside these organisms. And you know what they want to do? So basically E. coli will have its own genetic material. This particular virus's genetic material will try to get incorporated over here and use the E. coli's machinery to replicate. Now E. coli is so smart, it say that it cannot happen. I will stop this. How? Basically, as I said, it releases two enzymes. One enzyme which is known as the restriction enzymes, restriction endonucleases. Okay, the restriction enzymes, they are known as molecular scissors. They cut the DNA. So they go and cut the genetic material right there. Restriction enzymes cut it. But the same restriction enzyme can act on them also, their own genetic material. So what to do now? Basically, I have to tag myself and say that, hey, don't cut me. So for that, you need another enzyme which basically adds the methyl group. It adds the methyl group. So wherever methyl group is there, this restriction enzyme won't be able to cut. So two functions, one is to produce restriction enzyme, the second enzyme is the one that adds methyl group and says that, hey, you should not be cutting me. So that is the question. In the year 1963, two enzymes responsible for restricting the growth of bacteriophage in E. coli were isolated. One of them, they cut the DNA and the other one added the group, which is what? It is methyl. Okay. Now, going ahead with the next question. Question number 11. Which of the following is the correct representation of a primer annealing step in PCR? What is PCR? Polymerase chain reaction. And you are being asked which is the step where the primer is being attached correctly. So over here, if at all you see, first of all, you tell me, in polymerase chain reaction, you need an enzyme, that is DNA polymerase, to extend the DNA, to make a copy of the DNA. And in which direction does it work? It always makes a new strand in the direction 5' to 3'. That means the template strand which is there, it should be in 3' to 5' direction. Then only I can make a new DNA from 5' to 3', right? So obviously the primer has to attach in the 3' region. The primer always has to attach in the 3' region because the primer which is being attached over here in the 3' region, that is being elongated. That is being elongated by DNA polymerase in this direction, that is 5 to 3. Fine. So over here we have to find where do you have it attached. So 5' 3' is given and the primer is attached in the 3'. This is correct. But if at all you see this is wrong. And also both of these strands are in same direction. That cannot happen. One strand will be in 5' to 3', another one will be in 3' to 5'. Ulta. Okay, here 3' and 5' is there. In the 3' position, this particular primer is added. This is correct. But here it is not. It is added in the 5th position. So this is wrong. Or the 5' position, not 5th position, 5' position. Now over here again in the 5' position, wrong. Over here if you see, it's in the 3' position, and also on the other hand it's on the 3' position, which is why your answer for this question would be what? D. Okay, your answer for this question would be D. This is from Biotechnology Principles and Processes. Cool. Now let's go ahead with question number 12. Given below are steps in the gene therapy of a patient with adenosine deaminase deficiency. So gene therapy is given to patients with some deficiency of a gene. So what is this particular thing? Let me tell you first. So adenosine deaminase deficiency is caused because the gene coding for this particular enzyme, that is adenosine deaminase, okay, deaminase, is basically deleted. It is not there. Now when a gene is not there, obviously you cannot expect the product to be formed, that is ADA, adenosine deaminase. And why do you think this product is important for the cell? So basically ADA is there to remove the toxic substances from the cell. Now it is the same in all our cells. None of our cells have this, but it is of most importance for the lymphocytes. Lymphocytes are a type of WBCs. In lymphocytes, you have to make sure that the toxic substances are removed, otherwise it messes around with our immunity. So what to do now? If at all a person does not have this, can they stay for a longer time? No. Which is why you need to take gene therapy. What exactly is done over here? So some of our cells, suppose I have the problem, some of my lymphocytes are being taken. Now only some are taken because I need a few in my body also to survive, right? Some are being taken out and it has been grown in the medium. They are allowed to multiply in the medium. Now once they have multiplied, what do? Basically, I take the gene which codes for ADA. I take the gene that is coding for ADA and I put the gene inside all of these cells. So the cells that have been multiplied, first of all I took the cells and then I multiplied it within the culture medium. Now what I am going to do is that basically I am going to insert my gene within all these cells. Now after doing all of this thing, what will I do? Basically I transfer them back into the body, back into my body, and now I have the cells who has this gene to produce adenosine deaminase. But the problem with this is, it is temporary. You would have to do repeated transfusion. Why? Because the lymphocytes which are being inserted, they cannot live for long. They always have. They are not immortal cells. They are mortal cells. They will die after some time. Which is why you need repeated transfusions. Okay. Now here you are asked, what are the steps? Tell the steps in order, in the correct order. So first of all, lymphocytes are grown in culture medium outside the human body. This should be the second step. So I will mark it second, but I am not sure yet. I have to read the other option. Lymphocytes are altered with the functional ADA gene. Again, after culturing them, I am going to alter them as well. So this is the third step. Lymphocytes are collected from the blood of the patients. First thing is this only, I have to collect the lymphocytes from the blood, then basically I'll grow in a culture medium, then basically I'll alter with what? The functional ADA gene. Now, the genetically modified lymphocytes are infused back into the patient. This would be the fourth one. So your answer for this question would be 3, then 1, then 2, then 4. 3, then 1, then 2, then 4. Your answer is C. Your answer would be C. And always remember, you are going to put cDNA. You are going to put cDNA. What do you mean by that? Complementary DNA. So usually what happens is that the DNA which is there, it transcribes into an mRNA, and this mRNA gives information as to what proteins have to be made. So the DNA which is there, it has certain region which codes for proteins, it has certain region which does not code for proteins. So the ones that codes for proteins will be only found in the mRNA, because while this process is happening, lot of processing happens. A proper mRNA is given which just has the genes that is going to code the protein. Nothing else will be there, nothing extra would be there over here. Okay, it's pakka nice thing. Now, when you want to insert the gene, do you take it directly from the DNA? Because no, because my dear children, you will have both the coding regions and the non-coding regions. Which is why, what do? You basically make a DNA out of the mRNA. You do ulta step. You did like this, right? Now you do ulta. Make a DNA out of mRNA. And this complementary DNA which is being formed is known as cDNA. So that you just have the regions which actually codes for amino acids. Okay, this is known as cDNA. So if in your NCERT you have seen cDNA and you have been confused, I hope that confusion is clear. In our DNA, we have regions that codes for proteins, you call them exons, and there are regions that does not code for any protein. And this region which is there, it does not code for anything, it is known as introns. Okay, fine. Let's go ahead with the next question, question number 13. The organisms that reproduce only once in their lifetime is, okay, only once they are going to reproduce. What are these organisms? Example, oysters, pelagic fish, salmon fish, hippocampus. Your answer for this question is salmon. Now again, this is something that they have not asked much before, okay, but it is there this time, which means that they have entered into NCERT and then they're giving you questions from there. So I have taken it, the line exactly from your NCERT, I have just copied and pasted over here. Some organisms breed only once in their lifetime, Pacific salmon fish and also bamboo. So these two are organisms which only breed once in their lifetime, while others breed many times. Example, most birds and mammals. Okay, some produce a large number of small-sized offspring, such as oyster and pelagic fishes. They produce large number of small-sized offsprings, and some produce a small number of large-sized offspring, such as birds and mammals. Okay, so please remember that as well. It can be a question later on. So associated things are also given. Oyster and pelagic fish, they produce lot of organisms, and my dear children, they are small organisms, but they are a lot in number. Whereas birds and mammals which are there, they produce large-sized organisms, as you can see, and they will be lesser in number. Okay, so your answer for this question, for what organisms are the one which breed once, it is salmon fish and also bamboo if you have an option like that. Now this is from chapter organisms and population. Okay. Now question number 14. The decomposers especially abundant in the bottom of the pond are. The decomposers means the ones which feeds on the dead and decaying material. Now you are being asked specifically about the bottom of the pond. Again, something literally from within the NCERT. So we know this general, decomposers, they are what? They are basically FB, Facebook, right? Fungi and bacteria. Fungi and bacteria are the general decomposers. But since you have been asked about pond, in your NCERT there is a line given which you should definitely know. What is that? The autotrophic components, I'll come to this first. The decomposers are the fungi, the bacteria and the flagellates. Flagellates are also there. Okay, so it is flagellates, fungi and bacteria. Your answer for this question would be D. Let's see the other things. So what are the producers over here? The producers include the phytoplankton and some algae and the floating, submerged and marginal plants. So phytoplankton, some type of algae, and also the submerged or marginal plants which are there, they act as producers. Consumers are represented by zooplankton. Zooplankton feed on the phytoplankton. The free swimming and the bottom dwelling forms, that are the consumers. Now in the decomposers, you have these three things. As I said, you have to be careful here because the questions are coming from within. So yes. Now 15th question. These type of questions have come, but even though this can be traumatizing, because for me it's literally very difficult to remember the numbers like that. But still, my dear children, we will try to do so. So IUCN Red List 2004 documented the extinction of species in the last 500 years. Include what? 784. Extinction of 784 species, in which 338 of them are vertebrates, 359 of them are invertebrates, and 87 plants are there. You need to remember them also, the numbers there. So 338 are vertebrates, 359 are invertebrates, and 87 of them, my dear children, are plants. And in total, what is it? It's 784. This much extinction was there. Extinction of 784 species in the last 500 years. This is from the chapter Biodiversity and Conservation. Okay? Chalo. Now you have got fill in the blanks question, and this is my favorite part because this is super easy, right? To fill in the blanks, you have been given the options, you just have to fill it. So the initial few days of milk produced after parturition is, the initial few days, the milk which is produced, what do you call it? Once the baby is given birth to, that is what you mean by parturition. So initial milk is known as, from this option that you have, it is known as colostrum. This is what? Colostrum. And always remember, the colostrum is super rich in some antibodies. What is this antibody? IgA. IgA. A from Amma, you're getting the milk, right? So A, you remember like that. Now, oral pills are contraceptive, effectively function. Oral pill as contraceptive effectively functions by inhibiting what? So, oral pills are contraceptives, but how do they do their work? By inhibiting what? So, amoebiasis, richness, nostoc, ovulation, diversity. Ovulation is something that can easily fit in. Oral pills basically does not allow ovulation to happen. Now, if egg is not released, do you think the sperm can meet anything out there? No. So, your answer for this question would be what? Ovulation. This is the second one. Now, symptoms of constipation, abdominal pain and cramps associated with disease what? Constipation is there, abdominal pain is there and cramps is there. It is disease amoebiasis. Amoebiasis is caused by ant amoeba histolytica. It majorly affects, I have written over here, it majorly affects your large intestine. You saw all the answers, right? So anyways, it majorly affects your large intestine, do not forget it. So okay, I messed up with the spelling. Did I remove everything? It's okay. You have the answers in the next slide. Next one. Cyanobacteria is an autotrophic microbe. What do you mean by that? Autotrophic means they can prepare their own food. Cyanobacteria is an autotrophic microbe widely distributed in aquatic and terrestrial habitats, can fix atmospheric nitrogen is. So you are being asked which one among them is a cyanobacteria which basically can prepare its own food by fixing the atmospheric nitrogen. Fixing the atmospheric nitrogen means basically what they do is that they take in the different forms of nitrogen, that is ammonia and things like that, and they break them down into simpler forms which plants can easily take up. So this is what you mean by fixing of nitrogen. That means breaking down the complex compound into simpler forms, okay, and such that for other organisms it will be easy. So cyanobacteria is an autotrophic microbe widely distributed in aquatic and terrestrial habitats, can fix atmospheric nitrogen. Is what? It is nostoc. Nostoc is such a cyanobacteria. Okay, question number 20. Alexander van Humboldt observed that within a region, species dash increased with increasing explode area, but up to a certain limit. So basically he was voyaging and he found that this is a region, and when I moved in front, when I explored more area, species richness was increasing. There were many different species out there, but it happens only up to a certain limit, not more than that. Why? Because there is there is a problem with the resources which are there. It is limited, right? Which is why it's a problem. So over here, Alexander von Humboldt observed that within a region, species what? Species richness, okay? Species richness increased with increasing explode area, but only up to a certain limit. Cool. So your answer for this question is this, and this is the entire answers for all of the fill in the blanks question. Just give me a second, quick second. Okay children, let's go ahead with the next set of questions. Now this is the time for the 2-marker questions. So sexual reproduction in flowering plants is where you got this question from. Draw a label diagram of mature 2-celled pollen grain. Now that is easy, right? So how do you draw it? Over here, my dear children, always remember in the pollen grain, you have 2 cells. One is the bigger cell, which is known as the vegetative cell. V for very big cell. Okay, now this very big cell will have a nucleus which is irregularly shaped. The shape of the nucleus is also important. Irregularly shaped nucleus. Now within this big cell, you have a small cell which is floating. You call it generative cell, because it is responsible for generating 2 gametes later on. It also has a nuclei which is elongated in structure. Elongated nuclei. Now since this is a cell, you have the plasma membrane, but apart from that also you have certain layers. So one layer which is continuous, you call this layer as intine. You call this layer as intine because it is inside, and this intine is made up of 2 things, pectin and cellulose. Pectin and cellulose. So PC, Priyanka Chopra, you can remember like that also, okay? And now outside that you have another layer which is not continuous, it is discontinuous layer. And this discontinuous layer which is there, you call it as an exine. Okay, exine. An exine is made up of the toughest material out there which can resist alkali, acids, everything. It is known as sporopollenin. And whenever you do not have sporopollen, you call these gaps as what? Germ pore. You call this as germ pore. And why do you think germ pore is important? So basically, when the pollen grain land on the stigma, and if at all everything goes well, they have to form a pollen tube. And this is where the pollen tube comes out from. And who is the thing that helps in the formation of pollen tube? It's a vegetative cell. So vegetative cell is also known as tube cell. Why? Because it helps in the formation of the pollen tube. And then the generative cell will also divide into two. You have two gametes. These two gametes will be going through the pollen tube. Okay? So draw a label diagram. Please draw a proper diagram like this. It is going to be super helpful. You need not draw the vacuole. It's totally fine. But at least other things should be there. So tube cell, because it is vegetative cell, it forms the pollen tube later on. That's why it is known as tube cell as well. Cool? Fine. Now mention any 4 assisted reproductive techniques that can be advised to couples with infertility problems. Now since they have infertility problems, we have to go to infertility clinic, and there you have certain things such as IVF. What do you mean by IVF? In vitro fertilization. That means fertilization outside the body, within the laboratory condition. So over here, sperm and egg is taken and they are fused in the laboratory condition. This is known as IVF, where fertilization is happening outside. Now at the end of it, you will get a zygote or you will get an embryo. When the zygote is a single cell thing, embryo is what? Whenever the zygote divides, now you call this as an embryo. Okay, suppose you have the zygote. The second step that you do is ZIFT. Zygote intrafallopian transfer. Suppose the female who is there, who has everything which is perfect, her body is perfect for the conception process, just that there was some problem, fertilization was not happening. So what do? The zygote which is being formed, let's put it in the fallopian tube of the female. This is known as zygote intrafallopian transfer. Inside the fallopian tube. You are not going to put it in the uterus. Always remember, zygote is in the fallopian tube, it divides. The formation of embryo, and a type of embryo gets implanted into the uterus. Until then, it has to be there in the fallopian tube itself. Now, either you put the zygote, if you're putting the zygote, you call it zygote intrafallopian transfer. Now, suppose you're not putting the zygote, you are putting the embryo, which is more than 8 cell stage. Which is in more than 8 cell stage. That means this embryo is made up of more than 8 cells, suppose 16 cells. Then you do not put it in the fallopian tube. You will rather put it directly into the uterus. Okay, you will put it directly into the uterus. This is known as intrauterine transfer. So embryo with more than 8 blastomeres is transferred into the uterus. If it is less than 8 blastomeres, then you do ZIFT only. So it's zygote plus the embryo. So zygote up to 8 zygote and embryo. Zygote and embryo, which is up to 8 blastomeres, you can put it in the fallopian tube. If it is more than that, then you have to put it in the uterus. If you put this one in the fallopian tube, it can lead to ectopic pregnancy. That means pregnancy elsewhere, not in the uterus. And it can be dangerous for the mother as well as the child. Now, the next technique which is there, it is gift. Gift basically means that the parents are there. The female basically is not able to produce the ovum, but has all the other situations needed for conception to happen normally. That means her uterus and everything is fine. So in that case, why not I just borrow a ovum from someone else? Yes, we can do that. So that is known as gift. Gamete intrafallopian transfer. So while we take the gift, that is over, my dear children, you have to put it in the fallopian tube also, because that's where the sperm will come and do the fertilization. So fertilization is happening within the body of the female. You're just giving the ovum, that's all. So ovum from a donor is transferred into the fallopian tube of a woman with functional uterus for in vivo fertilization within the body. Now last one is artificial insemination. Even though you're asked 4, I have written 5, so that if at all you're not writing this, you can at least write 4 of them. Artificial insemination basically means, suppose male has some erectile dysfunction issues, the penis is not getting erected, or the semen is not being produced, or in the semen the sperm amount is very, very less. In that particular cases, obviously things are not going to work. So you need to do artificial insemination, where basically semen is being collected and artificially it is put within the female's body. It can be either in the reproductive tract, that is vaginal tract, or it can be within the uterus also. If it is within the uterus, you call it intrauterine insemination. If it is just normal, basically you do not have a term for it. In general, you say it artificial insemination. Okay, in general you call it artificial insemination. Semen from the husband or the donor is artificially introduced into the female reproductive tract. If you are specifically doing it within the uterus, then intrauterine insemination. Done? Done, ma'am. Next question, question number 23. Now this is something new, okay? So this is what I was talking about. Recombinants obtained in both Mendelian and Morgan experiment, yet processes involved are different. Comment. So Mendel's experiment, if at all you have seen in round and yellow and wrinkled and green plants, it showed that basically in the second generation I am getting round and yellow 9 of them, 1 of them is wrinkled and green, and 3 9 to 3 to 3 to 1, right? 3 which are there, they are new, they are round and green and wrinkled and yellow, something new. Non-parental combination is there. So that means recombinants are being found. Recombinants means new combinations is being found there. And Morgan also did his experiment and he said that I am also able to find new combinations out there. But both of them got it differently. How? What is the question? That is the question. Okay, so basically in the case of Mendel's experiment, the chromosomes that he was talking about, suppose I take this, capital R for round, small r for wrinkled, capital Y for yellow, small y for green. Over here when you do gamete formation, from this pair only one gamete is going to come, either this. So suppose it's this, then capital R. From here suppose it's capital Y, then you have got a gamete which is having capital R and capital Y. Next thing, from here suppose again it's capital R but here it is small y. You got another type of gamete. Now from here you got small r and from here you got capital Y. Another type of gamete. From here you got small r and from here you got small y, another type of gametes. Now when the gametes are going to fuse with another gametes out there from the other parents, what will happen? You will get newer combination which you didn't find before. What do you call this? This is independent assortment. The alleles for, the pair of alleles for one particular character is independently assorting from the other pair of character. So R does not say that I will go with Y only, capital Y only. Does it say? No. It is independent. From this only one has to go, from this one has to go. It can be either of them. This is known as independent assortment. And Mendel got different or recombinants out there because of this independent assortment. Now if you talk about what you call Morgan's experiment, my dear children, he got recombinants. Why? I'll tell you. So over there, suppose you have genes. This is having A and over here you had small b, over here you had capital A and here you had capital B, right? These genes which you see, they are far away from each other. Now suppose this piece got exchanged. This piece from here got exchanged. What will happen? You are getting a new combination where small a is now with capital B because it got exchanged, and here small b will be with small b, which is, capital A which is there, it will be with small b. Here also new combinations are being, now since the genes are different on the chromosome, my dear children, when the chromosome fuse, obviously you will get new things out there. So why is it happening over here? Because you are specifically focusing on chromosomes which have similar genes on similar location. I have the B genes, it can be capital B or small b genes at similar locations, here also. And here basically you are talking about linkage. Here you are talking about linkage. Linkage basically means, suppose this and this are very close to each other. So whenever things got exchanged, they both will together get exchanged. That means amount of recombination will be less, new combinants would be less. But if at all they are loosely linked, that means they are far away from each other, recombination can easily happen like this. It totally depends upon the distance between the genes and everything out there. So it is linkage or crossover that is happening over here. Rather here it is independent assortment, which is why you are getting new combinations. So in Mendel's experiment, recombination occurs due to independent assortment of genes on different chromosomes. Over here in Morgan's experiment, recombination occurs due to crossing over between linked genes. Linked genes, crossing over is happening on the chromosomes that have the same genes at the same loci. So it's not like I'm talking just about one gene, it can be two genes, but these two genes are homologous genes. Why? Because whatever I have over here, the opposite of that would be over here, or the similar of that would be here. These chromosomes are known as homologous chromosomes. So Morgan's experiment, it's because of crossing over, exchange of things that is happening, and over here linkage has a big role to play. and over here it's just independent assortment. Cool, I know this question is a little difficult to process in, but with time you will be understanding it better. Okay, just read through it once more. And if you still have doubts, you can ask me in the comment section. I will be there to help you out. Now, 24th question. Name the methodologies used in human genome sequencing. That means I have to sequence the whole genome. What was the technique that was being used? So you have two techniques. One is expressed sequence tag and the other one is whole genome sequencing, which is known as sequence annotation. So what is there in express sequence tag? Basically, we know the DNA which is there, it is transcribed into RNA. And the RNA is something that forms what? That gives information to form the proteins. Now suppose I just know the genes. I just, try to code the genes which are there on the RNA. Okay, the genes which, not the genes on the RNA. Basically, I just try to code the genes which are expressed as RNA or the ones which actually forms the RNA. That is known as expressed sequence tags, because I know they will form protein and blah 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 things. So only the genes expressed as RNA were identified and sequenced. So there are certain areas of the DNA which is not being expressed as RNA. What about them? They were not sequenced. Now in whole genome sequencing or sequence annotation, what is happening? The entire genome, whether it is coding, whether it's non-coding, all of them were sequenced and then the functions were assigned. Okay, so two methodologies, one is express sequence tag and other one is whole genome sequencing. Works? Going ahead. That is also a new type of question, and over here this is also a new type of question coming from evolution chapter. So name the evolution of any two plant forms through geological periods from tracheophyte ancestors. Name the evolution of any two plant forms through geological period from tracheophyte ancestors. So how do you frame this answer? So my dear children, first of all, know the picture very, very well. So just give me a second. So over here, you have some ancestors given. Chlorophyte ancestors, tracheophyte ancestor, Rhynia type plants, xylophyton and everything. You need to know the basic of plants first. So the initial plants which came up were thallophytes. Some things which do not actually look like the plants that we have today. They are thallophytes, okay, they do not have roots, they don't have anything. And then came the next organisms which were bryophytes, which had root-like, stem-like structures. Okay, so initially what did we have? I have to explain this, then only I can tell you all of this thing. So initially, just a second, we had something which is known as thallophyta. Okay, they didn't have root, stem, leaf, anything. They're like the algae out there. From thallophyte, we got bryophytes. Bryophytes are the one which had root-like, stem-like and everything like structure. But remember, they did not have a vascular system. That means there was no xylem, there was no phloem to transport food and water. And now from them came pteridophytes. From them came pteridophytes, from where you have got the vascular plants. That means they had what? The xylem, the phloem and everything out there. From pteridophytes, basically you got seed producing plants which are gymnosperms. They had naked seeds, which is why they are known as gymnosperms. Now from gymnosperms you got the flowering plants which is known as angiosperms. Over here the seeds were covered with the help of fruit and everything. This is how the flow goes. Now pteridophytes basically are also known as tracheophytes. Why? Because tracheid is a part of the vascular system of the plant, and the first organism or the first plant to have the tracheates is what? Pteridophytes. So over here, if we go, now if you see chlorophyte ancestors is there, that means you're talking about chlorophyte. That means the primitive ones, thallophytes. They are plant-like, they just have chlorophyll, that's all. That's why they are put under plants. So chlorophyte, from chlorophytes you got tracheophytes. Tracheophytes are what? Your pteridophytes only. From there you got Rhynia type plants, again the different form of pteridophyte. From them you got xylophyton. Now if at all you see, ma'am, from chlorophyte, how can you get pteridophytes in between? Bryophytes is there. So if you see this line, it goes above to give what? Bryophytes. In Cenozoic era, my dear children, there are a majority of bryophytes coming off from here. Now the Rhynia type plants, as I said, converted into xylophyton, and from xylophyton you got some modern day pteridophytes such as horsetail, such as ferns and everything. From here only you got some gymnosperms, okay, from gymnosperms, which is ginkgos, conifers, nettles as well as cycads. And now please understand, from here you got progymnosperms, from progymnosperms you got seed ferns, from seed ferns, my dear children, you got what? The angiosperms. So from gymnosperms you're getting the angiosperms, dicots and the monocots, which comes under angiosperms only. So here you have been asked about two plant forms. You have to tell about the two plant forms. So let's talk about pteridophytes first. So tracheophyte ancestors, from there the rhinotype plants, from there the xylophyton, from there you got ferns, and ferns is what? A pteridophyte. It's a pteridophyte. So you're talking about one plant form. The second plant form, angiosperms. So from tracheophyte ancestors, rhinotype plants, then xylophyton, then progymnosperms, then seed ferns, and then you got angiosperms. And then mention that appeared prominently from Devonian to Jurassic period. So over here they appeared prominently from Devonian to Jurassic period. This conversion happened from Devonian to Jurassic period. In the Jurassic period you had got the ferns. Now similarly you have to talk about this also, angiosperms also. So diversified mainly in the Cretaceous to Cenozoic era. So over here you have the Cretaceous era, from here they got diversified. In the Cenozoic era they were totally diversified and you got the actual angiosperms which were there. So please mention like that. You go through this particular picture also. Why is this question being discussed over here, such that you understand that this picture is also important. You need to go through it. And I think at least I could make you understand the basic part of the story, right? Let's go ahead. Now describe briefly the primary treatment of sewage. This is microbes and human welfare question. This is an easy question. So in sewage treatment you have the primary treatment, the secondary treatment and all of it. What is the primary treatment all about? It's simple. So in primary treatment you want the things which floats to be filtered out and the things that are sinking to be filtered out. So you have two techniques, one is filtration and the other one is sedimentation. So the floating debris whichever is there, it is being removed by the process of filtration, and whatever things are sedimented down which are of high weight, okay, it is removed through sedimentation. Now whatever is left, okay, whatever is left, the fluid which is left, you call this as effluent, and this effluent is the one that has been sent to the next treatment, that is the secondary treatment. So primary treatment of sewage is the first physical step in wastewater management, where large and small solid particles are removed. Floating debris is removed by filtration. Grit, sand, soil, pebbles, etc. is removed by sedimentation. The settled solids forms the primary sludge. Whatever is settled over here, you call it primary sludge and you remove it, okay? The clear upper liquid is known as effluent. The effluent is sent for secondary treatment. Simple, easy, right? Now 27th question, which I think is the end of 2-marker question. Let's see. Differentiate between ex situ and in situ conservation. Ex situ means you are conserving it outside their natural resource. In situ means basically you are conserving them in their natural habitat. So that is the first difference. Conservation of species in their natural habitat is in situ. Conservation of species outside the natural habitat is ex situ. Now protects the entire ecosystem. So whenever you are conserving the species in their surroundings, basically you are going to make sure that the entire ecosystem is also conserved, because they have to sustain there only, right? So that is one big thing. Over here in ex situ conservation protects only the selected species, none of the other things are being protected out there. Examples. So wildlife sanctuaries, national parks comes under in situ conservation, whereas zoos, botanical gardens, seed banks, okay, comes under ex situ conservation. Fine. And so whereas wildlife parks also, that comes under ex situ conservation, okay, please remember that. Wildlife sanctuary is different, it is in situ, but wildlife natural parks which is there, that is coming under ex situ conservation. That's all. Yes, I was correct when I said that now comes the 3-marker question. So over here the first question that is usually being repeated is draw a neatly labeled diagram of the monocot embryo. This is from sexual reproduction of flowering plants. And how do you draw the easy diagram of monocot embryo? First of all, know the basics. So in the monocot embryo, obviously you need an embryonal axis. So this is the embryonal axis. This is going to be your plumule, which forms the shoot. This is going to be the radicle which forms the root, and then the radicle has a root cap. Now it's a monocot. It's a monocot, which is why it just has one cotyledon. And you know what? Since you just have one thing, you name it properly, you call it scutellum. This one cotyledon which is there, you call it scutellum. Apart from that, you also have some endosperm which is still persisting out there. Some endosperm is still persisting over here. Now the plumule which is there, it is surrounded by something which is known as coleoptile. It is known as coleoptile, that protects the plumule. Over here, the root cap is also protected by something which is known as coleorhiza. So coleorhiza has an R, over here root cap also has an R, plumule has a P, over here coleoptile also has a P. So this is the basic thing that you need to remember. Now how do you put it in the picture format? So you need shoot apex, that is a plumule covered by coleoptile. You need radical, root cap and coleorhiza. You have one cotyledon which is known as scutellum. Endosperm is not there in the picture. What is this epiblast? So there is formation of one more cotyledon, but it is not able to form itself. So it is rudimentary. It does not form. It is known as epiblast. Now how to come from this picture to this picture? So for that, we need a bit of the practice. So over here, make a big mouth of a whale. Okay? And now this whale has two hands. One hand goes like this. And now you go ahead and make a tail. So this is the tail. And now you make a small hand. Cool. Now my dear children, over here make a V structure like this. This is going to be your coleoptile. Again make a structure similar to that. This is going to be your plumule. Right? Now you connect this, make something like this, bring it over here, go to one tail and just put it abruptly up there. This is your radical. There should be someone to cover it. This is going to be your root cap. And whatever extra portion is there is going to be your coleorhiza. This is going to be your epiblast. This is going to be the scutellum. And my dear children, what do you have? This is going to be your coleoptile. And this is going to be your plumule. If at all the question is draw a neat label diagram of monocot embryo or grass, anything, just draw this. You'll get your marks. Okay? Yes. Now, question number 29. What is menstrual cycle? Mention one pituitary and one ovarian hormone that plays an important role in menstrual cycle. So first of all, what is menstrual cycle? It is a cycle. Okay, it is a monthly cyclic changes that occurs in a female's body, especially in the ovary and the uterus, and it is going to happen in a gap of every 28 days, or basically the cycle is of 28 days duration. So menstrual cycle is a monthly cyclic changes in the ovary and the uterus that occur in the human females for about 28 days, starting from menstruation to the next menstruation which is there. Now you have been asked to mention one pituitary and one ovarian hormone. So pituitary hormones are two. One is FSH, follicle stimulating hormone, and the other one is luteinizing hormone. The ovarian hormones are also two, that is one is estrogen and the other one is progesterone. Okay. Now FSH, if I take that up, what does it do? Follicle stimulating hormones develops the follicles or makes the follicles mature. So follicle stimulating hormone stimulates the growth and maturation of the ovarian follicles in the ovary. So in the ovary, these follicles, this FSH comes and acts on the follicles, making it mature from primary to secondary to tertiary to Graafian. And while it does, you know what, I told already, the follicles are made up of granulosa cells. These granulosa cells are going to release some estrogen, and that estrogen is very much important in our uterus. So this estrogen goes to our uterus and helps in the formation of a layer which is known as endometrium. Okay, so FSH, which is a pituitary hormone, is indirectly helping in the formation of estrogen. So it induces the follicles to secrete estrogen as well. If we talk about estrogen, it causes repair and proliferation of the uterine endometrium after menstruation. So in menstruation, the endometrium goes down, right? Now to reform it again, you need estrogen. And it also leads to LH surge causing ovulation. So basically, when the amount of estrogen goes up high, what usually happens is that LH and FSH, both of their level also goes high. And when the LH has the peak, okay, it is known as LH surge. The follicles which are generating, the Graafian follicle which is there, it ruptures to give the secondary oocyte. So the picture, it looks like this, my dear children. So over here, during menstruation, the level of estrogen and progesterone, both of them are less. Okay, which is why what is happening? Menses is happening. The endometrium is getting broken down. Now on the other hand, In this situation, the follicles will be generating. Why? Because you want the next cycle to begin up. The follicles are generating. Now when the follicles are generating, the follicles will be under the influence of FSH and LH, majorly FSH. What will happen? It will release some estrogen. Now since estrogen is there, what will happen? Slowly the endometrium will start to regenerate. When the estrogen reaches a peak, what happens? Over here, FSH and LH also reaches a peak, especially LH, which is why you have a rupture of follicle, and that's how the secondary oocyte is being released. Now when the secondary oocyte is being released, whatever is left behind is known as corpus luteum. Now corpus luteum releases another hormone, progesterone, which is why the level of progesterone goes up high, and that's how the endometrium becomes even more thicker. That is the basic thing. So I've been asked about FSH, or basically any pituitary hormone, and also the ovarian hormone. Please explain your answer properly according to this. Okay? So over here you just have to mention, but it's not like that. Please explain also. It's a 3-marker question. Now question number 30. Write any 3 principles to be followed by people to be free from sexually transmitted infections. So what are the 3 principles that need to be followed to make the people safe from sexually transmitted infection? First thing, avoid multiple partners, because that leads to more chances of STDs. Second, correct and consistent use of condoms, because condom is the only contraceptive which prevents conception and also prevents sexually transmitted infections. Why? Because when you wear a condom, there is no genital fluids that is being mixed. There is no blood which is being missed. It is being covered completely. Okay. So the correct and consistent use of condoms, and prompt medical checkup and treatment. That is also there. If you do prompt medical checkup and treatment, my dear children, you will be more aware. You will be making sure that you should not be doing this thing out there. So 1, 2, 3 and there. The 4th one is an extra thing, avoiding sharing needles and razors. So not all sexually transmitted diseases, my dear children, are necessarily transferred from the blood, which is why this is an extra point. This can be put down, but majorly it's these 3 points that you should be putting up. Okay? Now 31st question, this is related to evolution. Draw a label diagram of Stanley Miller's experiment in relation to chemical origin of life. So this is the experiment which was being done by Stanley Miller. Over here you can see boiling water is there. This basically sends out vapor. This sends out vapor because vapor was there in the early atmosphere. And over here you have electrodes. When the electrodes basically they are on, they have the effect of lightning. So we have believed that lightning effects were there. Because of the lightning, so many reactions happened up over here. That is what we believe. And also it is responsible for temperature. So 800 degrees Celsius is being maintained over here. And as you can see, it is completely closed. There is no oxygen. This is anaerobic condition which is being maintained. And you have certain materials out here. Okay, CH4, that is methane, NH3 ammonia, H2O water, and H2 is there, hydrogen is there. So these are the 4 components that you need. What will happen? These gases in the presence of the lightning effect by electrodes and also 800 degrees Celsius and anaerobic condition, it is basically going to form organic compounds. So this inorganic things, okay, inorganic gases out there, they'll come out in different combinations to form organic compounds, and that is what you can collect from here. In the rock bottom, it will be accumulated, and this was found that okay, you do not need the entire thing. Basically, in the presence of these gases and in an atmosphere which is anaerobic condition, you can produce organic things, and these organic substrates probably come out in different combination to form the RNA, DNA, all of it, and that's how we have evolved. So this is the Stanley Miller's experiment, and you have to draw the diagram like that. Cool. Now 32nd question. This is from Human Health and Diseases. Name the causative organism of the following diseases: filariasis, ringworm, and malaria. Filariasis, my dear children, also known as elephantiasis. Over here, you have a an ascalminthus. Okay, an ascalminthus, which is the causative agent. It is known as Wuchereria bancrofti. There is another version of it. Wuchereria malai is also there. But anyone you can mention, or if at all you want, you can mention both of them. Now ringworm, even though worm word is there, it is actually a fungal disease. It's caused by trichophyton, epidermophyton, and all of it. Microphyton, epidermophyton, trichophyton. So you can write all of it. Okay. Now malaria. Malaria is caused by not the mosquito. It is caused by Plasmodium. And in Plasmodium, again, you have different types. Plasmodium vivax is there, falciparum is there, P. malaria is there. The most malignant form of malaria is being caused by P. falciparum. Cool? Cool. Now, question number 33. So that was a direct question, which is why I didn't explain much. So question number 33 is there, mention the uses of following bioactive agents, streptokinase, cyclosporin A and statins. Now streptokinase, basically it is going to act like a clot buster. So you might have some blood clots, and these blood clots might lead to what? Heart attack. So to what do you call, burst the clots out there, you have some clot buster, and streptokinase is one such clot buster. Cyclosporin A, my dear children, it's an immunosuppressive agent. For example, I had an organ transplantation. Now my body feels that something foreign has come up, which is why my body will react to it and might kill the new cells or the new organ that I have taken up, and I won't be able to survive then. So which is why you need immunosuppressive drug as well, such that it suppresses down my immune system whenever I have something like this. So cyclosporin A is an immunosuppressive drug. During organ transplantation, it is being used. Now statin. Statin, basically you can remember, like statin stabilizes your blood cholesterol level. It actually lowers down your blood cholesterol level and leading to what? The heart diseases. So used as cholesterol lowering drugs to reduce blood cholesterol and prevents what? The heart diseases as well. So these are the three things you need to remember. This is from Microbes in Human Welfare. Now question number 34, this is from Ecosystem chapter. Now describe the steps of decomposition that operate simultaneously on the detritus. Detritus is what? The dead and decaying substance. So you have to tell, basically it is being asked about the steps of decomposition. So first thing is fragmentation, where basically what? A group of bacteria which are there, basically not bacteria, sorry, the detritivores which are there, for example, earthworm and everything, they are going to break big things into smaller things. So big chunks are being broken down to smaller things, which you call as fragmentation. What do you call this as? Fragmentation. Now when you break things down, usually what happens, some things which are easily, what do you call, can be absorbed by the soil will be taken up by the soil. This is known as leaching. So water soluble nutrients, whatever water soluble nutrients are there, they are dissolved and washed down into the soil. That is what you mean by leaching. Now the third thing, catabolism. So we have broken down the bigger thing into smaller thing. Whatever water soluble things were there that moved down into soil, that is known as leaching. And now, my dear children, the things which are there, the smaller fragments which are there, they have to be broken down again. This time not by detritivores but by decomposers. Okay, that is known as catabolism. Bacteria and fungi secrete enzymes to break complex organic material into simpler substances. So complex organic material which are there, they are broken down to simple inorganic. Always remember, simple comes with inorganic, complex comes with organic. Okay, so complex organic material are being broken down to simpler substances out there, but always remember, this is not a one-step process. Okay, so basically suppose I have a complex organic material over here, it is broken down to simpler material. In between you get something which is known as humus, which is like a partially decomposed thing. So that is the next term, humification. Formation of humus, a dark amorphous colloidal organic matter in the soil. So basically humus is also being formed, which is known as humification. Now later, this humus, my dear children, from here to here, this humus is being broken down again, and that's what you call as mineralization. So humus and organic matter are converted into inorganic nutrients like nitrogen, phosphorus, etc. So as I said, catabolism means A to B. But in between that, you have humification, a humus is being found, and then this humus is being broken down to what? Or basically humus which is there, it is converted to inorganic nutrients such as nitrogen and phosphorus. So these are the steps, 5 steps, you have to remember all of them. Fragmentation, leaching, catabolism, humification and mineralization. And you might be asked the order also, make sure you go like this itself. Cool? Now, we are getting ahead into the 5 marker session. Draw a neatly labeled diagrammatic sectional view of the human male reproductive system and label the given parts: testis, seminal vesicle, glans penis, epididymis and urethra. So over here the sectional view is asked. In your NCERT the sectional view is different. Just inquire from whichever teacher you have which diagram to draw, but in general, even though sectional view is asked, this is a diagram which is being followed. So this diagram has to be made and all of their parts have to be mentioned. Now if at all you are someone who is facing difficulty in making the diagram, I can help you with that. So over here you have to make the testes. So I'll make it on one side. Okay. So my dear children, what exactly happens over here is that you have so many testicular lobules, right? Testicular lobules has so much seminiferous tubules. 1, 2, 3 seminiferous tubules are there. The seminiferous tubules moves outside, you call this and form a structure, you call this tubuli recti, but that is not given in your NCERT. You will directly come to a structure which is known as rete testis. It opens up into something which is known as rete testis. Now the rete testis try to exit out, which is known as vasa efferentia. This is known as vasa efferentia. E for exit, you can remember like that. But they are not able to exit, rather they form something like this, which is known as epididymis. Epididymis. Okay. Now over here I have to form the urinary bladder. Let's make the bladder first. So this is the urinary bladder. Okay, and this is the urethra. This is the urethra and this is the urethral meatus, the opening of it. Now you might be wondering, why did ma'am make something like this over here? It's not meatus, it is meatus. So I'll write the spelling also. I'm not okay. Anyways, fine. And here it is connected to the ureter, which comes from the kidneys, right? So now the epididymis which is there, it now opens into something else which coils up like this. This something else is known as vas deferens. So rete testis, vasa efferentia, epididymis and then vas deferens, which coils around the ureter and comes like this. Now over here it receives a secretion from a gland which you call as seminal vesicle. The same gland is on that side also. I'm just making one side. Together now they are forming a duct which is known as ejaculatory duct. This is known as ejaculatory duct together. Now the ejaculatory duct is going to open into what? The urethra. So from that side also you get the ejaculatory duct. Always remember over here you have an unpaired gland. This unpaired gland which is there, it is known as prostate gland. And from here you have the penis being starting. And just above you have another gland which is paired just like seminal vesicle. It is known as bulbourethral gland. Bulbourethral gland, you also call it Cowper's gland. So 3 glands are there, this. Now the penis is there. At the end you have glans penis, the enlarged portion of the penis, and it is being covered exclusively by a skin which is known as foreskin. Okay. So this is a diagram that you need to draw, and that would be more than enough. You need not draw this big urethra. I just explained, I just wanted to explain you, which is why I made it like that, okay? Okay. So make a diagram like that, and this will be done. I've given you a hint. Go through it properly. Going ahead with the next thing. Represent schematically the results of 2 gene inheritance interaction by taking seed shape and seed color in pea plants. So basically you are being asked about the dihybrid cross experiment. Okay, so represent schematically the results of 2 gene interaction by taking seed shape and seed color. Seed shape and seed color, my dear children. So seed shape, suppose I have round, and over here I have wrinkled plant. Okay, and seed color I have yellow, and over here I have green. How do I represent it? First of all, I represent the pure breed. So capital R, capital Y, capital Y. Here small r, small y, small y. Now when you make a gametes, my dear children, always have this in your head. Always. This thing which is written like this, it looks like this. So from a pair of allele, you're going to get 1. From this, you're going to get 1. Anyway, it will be capital R. From here also, anyway, it will be capital Y. Same thing. small r and small y. All the gametes will be same. There will be multiple gametes which are formed, but all of them are going to look the same. Now when you cross them, what do you get? You get capital R, small r. You get capital Y and small y. So basically if I look, it looks like this. Capital R, small r, capital Y, small y. This is what my F1 generation. All of them are going to be what? Round and yellow. Why? Because R is dominant over small r and Y is dominant over small y. Now you do the selfing again, because you are being asked for that only. You have to talk about everything. So when you do that, so capital R small r, capital Y small y, capital R small r, capital Y small y. This is the new thing that you have got. Now again you have to make a picture like this for your understanding. So capital R small r, capital Y and small y. From this pair one can come, suppose capital R is coming, from this pair capital Y is coming, suppose. Then this gamete is going to look like this. From here capital R is coming, from here small y is coming, then it looks like this. From here small r is coming, from here capital Y is coming, then it looks like this. From here small r and from here small y is coming, then it looks like this. So 4 type of gametes from this parent, 4 type of gametes, similar type of gametes from that parent. You cross them. You make a Punnett square like this and you cross them. And that's how you are going to show schematically the results of 2 gene interaction by taking seed shape and seed coat color in the pea plant. Okay? I hope you understood. Going ahead with the next question, 37th question. DNA is genetic material in both prokaryotes and eukaryotes, yet the packaging process in their cells is not the same. Explain. So DNA is genetic material, we all know, right? But in prokaryotes such as bacteria and in eukaryotes, things are kind of different. Why? First of all, prokaryotes as in bacteria, they are small. The genomic content will also be small. In eukaryotes as us, we are advanced, which is why our genomic size is going to be very much. Second thing, they do not have nucleus. They do not have any cell organelles that are surrounded by membranes. They are prokaryotes, primitive structures. Nucleus is not there, which means the DNA is just floating in their body, right, in their body. And they do not need any histone proteins and things like that. But in the case of us, the eukaryotes, the DNA is long and we have to fit it inside the nucleus. Which is why we need help of some histone proteins. Okay, so that is the whole thing. How do you need to frame the answer? DNA is a genetic material in both prokaryotes and eukaryotes, but its packaging differs due to differences in genome size and also cell organization. In prokaryotes, DNA is naked, circular and present in the nucleoid region without histones because of the small genome and absence of a nucleus. Okay, there is no histones. Now in eukaryotes, DNA is associated with histones. About 200 base pairs of DNA wraps around a histone octamer. That means 8 histone molecules come together and the DNA wraps around that twice. So that 200 base pairs are covered here itself. This whole structure is now known as a nucleosome. Similarly, you will have another nucleosome. The wrapping continues, my dear children, such that it is compact. So these nucleosomes coil further to form chromatin. The chromatin condenses again to form the chromosomes. So thus that the large amount of DNA is packaged into the nucleus. You can give the examples of prokaryote, that is E. coli. E. coli's genome size is what? 4.6 into 10 to the power 6 base pairs. 4.6 into 10 to the power 6 base pairs. Ours is what? 6.6 into 10 to the power 9 base pairs, my dear children. So you should know that. Okay? Next, question number 38. Differentiate innate and acquired immunity. Mention the cells involved in accomplishing the acquired immunity. Refer to the below given diagram and answer the questions. Name the molecules trapped in the lymph nodes responsible for activation of lymphocytes that bind to A and B. Okay? We'll go question by question. Differentiate between innate and acquired immunity. Innate immunity is something that we have got from our birth. It's there with us from our birth, such as skin, tears, everything. Acquired is something that we are acquiring through our lifetime. Probably, suppose I have got an infection and now my body is reacting to certain things, producing certain cells. That is acquired immunity. Or probably I take vaccination, which is why my body is producing certain cells, giving me immunity. That is also acquired immunity. So acquired immunity is something that you get during, if at all you have an infection or you underwent what? Vaccination. Now, innate immunity, my dear children, it is non-specific. That means suppose tomorrow a bacteria comes, a virus comes, my skin is not going to allow either of them. It's not specific. Now acquired immunity, basically I know that for this thing I have to produce this. So it is kind of specific. Okay, specific to particular antigens. Antigens means foreign substances. It is specific to whatever foreign substances are going to come through. There is no memory. My skin does not remember that a bacteria came through. Whereas when you talk about acquired immunity, as I told, cells are being produced. There is some secretions happening. So basically certain cells are there which are memory cells. So they know that in this infection I have to release this. These are known as memory cells. So has immunological memory, not memory as you think, but whenever a reaction happens, there are two groups of cells being produced. One cells which goes and fights, another cells which remembers. They are memory cells which are being produced. Now the next time something comes up, they will be initiating the process or doing distribution. Example, skin and the phagocytes. Example, antibodies and the T cells. Okay, in acquired immunity, you basically have two cells which plays a very important role. So that is the next question. Mention the cells involved in accomplishing the acquired immunity. So there are two cells, one is B lymphocytes, another one is T lymphocytes. B lymphocytes produce the antibodies, and T lymphocytes basically are responsible for giving information to certain cells that you have to do this, you have to secrete this, you have to kill the cell and all of it. So there are different type of T cells, T helper cells, T cytotoxic cells which directly goes and kills, okay. And T cells, my dear children, are the major ones which are involved in graft rejection. For example, I have an organ transplantation being done in my body. So this is being rejected because of T cells, because T cells know that this is foreign, I have to remove it. Okay, so T cells are also responsible for providing immense amount of immunity. Now name the molecules trapped in the lymph nodes. Lymph nodes are what? Lymph nodes are lymphoid organs. Lymphoid organs are places where either the lymphocytes are being produced or they are maturing, or basically where they go and do the action. There are two types of lymphoid organs. One is the primary, where they are produced and matured. One is secondary, where they do the action of doing distribution. So lymph node is a secondary lymphoid organ. You have been asked, name the molecules trapped in lymph nodes, which are trapped in lymph nodes, which is responsible for activation of lymphocytes that binds to A and B site. That A and B site is what? You should know A and B site. So this is the picture over here. A and B site is given, and they're saying something is getting trapped and going to basically bind over here. The trapped thing is the one that binds over here. What is this? This is an antibody. An antibody, this site is known as antigen binding site. So this thing which is there is antigen, the foreign particle which is being trapped. Okay, always remember that. So A and B are nothing but the antigen. Now what does H2L2 in an antibody represent? So over here H2 is given. It's not given, but I'm telling you. These two strands which are there, these are H2, heavy chains. How many of them are there? There are two. Similarly, you have two light chains. That is why L2. So H2 and L2 represents the light chain and heavy chain of an antibody. Okay, H2L2 represents heavy and light chain of the antibody. That's all. Now we're going ahead with 39th question, which is this. Explain the process of preparation of competent host for transformation with recombinant DNA. So process of preparation of competent host means what? Basically, I have to make an organism ready to take up the new DNA out there. So what is the process that is being done? That is the question, first question. So we'll go ahead with the first question and then we'll come back over here, okay? So first is, suppose it's a bacterial cell. Okay, suppose it's a bacterial cell. So we can do one thing, that is we can treat the bacterial cells with divalent cations such as CaCl2. Okay, CaCl2. Calcium basically acts as a divalent cation over here. The Ca ions neutralizes the negative charge. DNA is negatively charged. I hope you know that. So it neutralizes the negative charges of the DNA, because why? We basically want the DNA to enter inside the cell. So if it is keeping on that negative charge, it might not be allowed to get entry into the cell. So first of all, the divalent cations which are there, they neutralize the negative charges on the DNA, which has to be picked up, and also on the cell membrane, the cell which is going to undergo this process. Now the mixture of cells and DNA is then given a heat shock. So initially it is in a cold environment. Now we are putting in a heated thing. Okay, heat shock is given. So the temperature is being shifted. And now this temperature, when it is being shifted, my dear children, what happens? There are transient pores. There are temporary pores that are being opening up. And because of this temporary pores, what will happen? The DNA which is being mixed, it can easily enter. So basically, I have a mixture of DNA as well as the cells. And now when they have pores, they will be taking up this particular DNA inside. Okay, I have neutralized both of them with the help of divalent cations. Now again, I put it in the cold. Why? Because I want to close this up. Okay, they close this up. This creates temporary pores allowing recombinant DNA to enter the cell. So this is one method of preparation of competent host for what? Transformation. Fine. Now there is the next question given to you guys, and that is refer to the given diagram of PBr322, which is a plasmid, and answer the following question. Mention the restriction enzymes that can be used for insertional inactivation of antibiotic resistance. Now, you have been asked, you are told that what are the enzymes that I can use for insertional inactivation of antibiotic resistance. That means if I am inserting something out there, the antibiotic resistance should be gone. So there are two antibiotic resistant genes, one is ampicillin and the other one is tetracycline. Okay, and over here, this is also the site for restriction enzyme BAMH1 or SALVin. Suppose BAMH1 makes a cut over here and inserts something else, what will happen? It won't be tetracycline resistant anymore. Similarly, if I use PVU1 to cut this to insert something over here, my dear children, it won't be resistant to ampicillin. So you have been asked, what are the restriction enzyme I should be using for making them insertional inactivation happening up there. So when I am inserting, things should be getting inactivated. So either I can use BAMH1 or SALVin for tetracycline then, for insertionally inactivating tetracycline resistant gene, or I can use PVU1, PST1 for insertional inactivation of ampicillin resistant gene. That is the question. So you have to tell about them or them. For that you need to know the location. If at all you do not know the location, what will happen? You would not be able to answer this question. So mention the restriction enzymes. I hope you got the restriction enzymes name. Now how many cloning sites are present in the given PBr322? Cloning sites means where I can fit in my things. So this, wherever restriction enzyme is there, that many cloning sites will be there. So 1, 2, 3, 4, 5, 6, 7, 8. 8 cloning sites are there. You have to mention about all of them. Okay, so the restriction enzyme used for insertional inactivation of antibiotic resistant gene, BAMH1, SAL1 for inactivating tetracycline gene, and PST1, PVU1 for inactivating ampicillin gene. Okay, now number of cloning sites in PBr322, 8 unique restriction sites are there, and you have to mention all of them. Now going ahead with the next thing, question number 40. That is, genetically modified plants have been developed through core techniques of biotechnology. How are the genetically modified plants useful to humans? So human beings, suppose a farmer is there. If at all I am producing a GMO plant, that is genetically modified plant, it will be so easy for me. Why? Because first of all, it has efficient mineral usage. That means even if I give little amount of minerals, even if I put little amount of minerals in the soil, still the plant will be able to survive. Why? Because it has a good mineral usage, efficient mineral usage. That is one thing. So plants grow faster, use nutrients better and produce more grains of fruits. This helps farmers get higher productivity even from limited land and also limited nutrients. Second thing, resistance to pests and diseases. Suppose I'm having a genetically modified plants which is resistant to insects such as Bt cotton. Okay, or I produce plants with the help of RNAi technology, that is RNA interference technology. In that, the plant will be resistant to Meloidiga incognitia. Then, basically, I am getting resistance, and that's how I can increase the productivity again. So, resistance to pest and disease is the next step out there. And you can give example as I have given over here, if at all you wish. Now, reduced use of chemical pesticides. This is the next thing. Okay, reduced use of chemical pesticides because, my dear children, they are pest resistant already. And tolerance to abiotic stresses. They can survive even in a condition where drought is there, where there is no enough water or where there is too much salinity. They can be surviving up there because they are resistant to that. Now, improved nutritional quality. This is one of the major things. So, basically, in the case of plants, suppose I am, so in the case of Africa, people were starving and they were dying because of malnutrition. But this problem was sorted because the major staple food was rice. Suppose I am producing rice which already has vitamin A in it. Okay, I am producing the plant which makes the rice which has vitamin A in it. Don't you think the plant is of nutritional quality and it gives, additional benefit to the people? Yes, so it was done, and that's how you made the golden rice. So golden rice was produced with the help of this. So genes can be added to increase vitamins, minerals as well as protein. So golden rice enriched with vitamin A, this helps to fight malnutrition as well. Now 6 says it reduces post-harvest losses. So genetically modified plants, usually what happens is that when we store, the things get lost. Okay, some pest again will come and eat it, and their longevity is very less. The seeds might basically become bad later on. But over here, they are more resistant to all these things. They have more shelf life as well. Okay. So these are the points that you need to write. Now question number 41. Write an equation to calculate the population density at the time t. So the equation goes like this. Nt is equals to n naught b plus 1 minus d plus e. So over here nt is the the population density at time t. N naught is the initial population density. B means birth rate. I means immigration. Immigration means amount of organisms coming into a population. Plus minus d means death rate, and e means emigration, people going out. So in total, this is the formula, and this is what you need to write. Now population density fluctuates due to change in 4 basic processes. Mention and define the 4 basic processes. So natality, that is the birth rate. It is obviously going to change the number of individuals added to the population by reproduction per unit time. Mortality, the death rate, the number of individuals lost from a population by death per unit time. Immigration, the number of individuals entering a population. Emigration, the number of individuals leaving a population. All of them put together, you have to give the answer. Okay. Now you have the second set of 5-marker questions, but over here basically you just have to choose one. Okay, you just have to choose one. So complete the given tabular column with respect to male and female gametophyte of angiosperms for the mentioned character. So number of cells present in the mature male gametophyte and also in the mature female gametophyte, and then all this thing. So first thing, what is the male gametophyte? It is the pollen grain. You should know that. Pollen grain is the one that forms the gamete, which is why it is known as gametophyte. Over here, the female gametophyte is nothing but the embryo sac. And if at all you know the structure of embryo sac, my dear children, it looks like this. There are 3 cells. Just a second. And do we have the picture? No. I do not have the picture, which is why we need to make it. So there are 3 cells right at the top. They are known as antipodal cells. All of them, they have their nucleus. Okay. Now on this side, on the other end, my dear children, there are other cells. 2 of them are known as synergids, and their best friend, the egg cell, which is the gamete. Now usually all of them are going to be haploid in nature. All of them are going to have half the number of chromosomes in nature. But if at all you see this cell, which is the largest cell in between, my dear children, it basically has 2 nucleus, 2 polar nuclei there. So N, haploid plus haploid, they are diploid in nature. The central cell which is there, it is diploid in nature. Always remember. So number of cells present in gametophyte. Male gametophyte, 2 cells, vegetative, generative. Now this generative can divide to form 2 gametes also. So 2 gametes plus vegetative, 3 cells are there. So either it can be 2 or it can be 3. 2 or 3. Now mature female gametophyte, in that how many cells are there? 1, 2, 3, 4, 5, 6, and this is the 7th cell. 7 cells are there. Now number of gametes present in. So gametes are 2 in number in the case of males, but there is only 1 gamete in the case of females. Type of cell division involved in the development of pollen grains. So how do you think pollen grains are developed? It develops with the help of mitosis. The cells divide, right? That's why you get 2 cells out there. So mitosis. How about over here in the case of female gametophyte? Here also mitosis happens, and that's when you get all of the cells. So I'll write mitosis. Now ploidy of the cells. What is the ploidy? All of them in the case of pollen grain, vegetative cell as well as the generative cell, they are haploid in nature. So over here all of them are haploid. But what about here? All of them are haploid except the central cell. Central cell will be diploid. Rest all of them are haploid. So you write that. So over here I have explained it properly. Number of cells present 2 or 3. 3 when you have 2 male gametes. Here it is going to be 7. Number of gametes present 2 male gametes, over here 1. Type of cell division involved in the development of mitosis. Over here also mitosis, and I have explained from microspore to this. Here you have to tell from megaspore to this. Ploidy of the cells, it is haploid, but over here it is haploid. All the cells except central cell, which is what? Diploid. Now you have a second part of the question which says, which advantage offered by seeds help angiosperms to adapt to changing environmental conditions? Angiosperms means flowering plants. So what are the offerings given by the seed that helps the flowering plants to survive in under different environmental conditions? So you have to tell that. First, seed, my dear children, will be having seed coat, which is testa and tegmen. And this seed coat protects the embryo out there which is inside. Basically, the genetic material is being saved all because of the seed coat, and also it provides dormancy. So when the plant does not have, seed does not have any favorable condition at that particular time, it is going to stay dormant because it has a protective coat around it. Seed coat protects the embryo and allows it to remain dormant during unfavorable condition. That is first thing. Second, food for the embryo. So food for the embryo is also there in the seed in the form of either endosperm or in the form of cotyledon. Dispersal. Seeds are adapted for dispersal by wind, water or animals, helping the plants to spread to new areas. So plants, embryo is being safe, everything is being safe, and that's enough. That is the advantages of having a seed. Okay, it is totally covered and protected. Now, principles of inheritance and variation. Mendelian disorders may be autosomal or sex-linked, dominant or recessive, homozygous or heterozygous. Pedigree symbols are used to analyze these disorders in human population. Mention the genotypes for the following pedigree symbols. Autosomal dominant. Autosomal dominant. So basically, my dear children, autosomal dominant disorders are the ones which will be appearing even if one is dominant, one allele is dominant. So over here it can be capital A, both the alleles are dominant, or it can be capital A, small a. Even if one is dominant, you will get something like this. Here, autosomal recessive. So autosomal recessive means they are autosomal chromosomes, but both of them has to be small letters, recessive in condition, then only you will be having this thing. So autosomal recessive, over here it is normal. Here it is normal that is given. So if it is normal which is being given, my dear children, you need to understand this is disorder is autosomal recessive, but here the female is normal. So that means you cannot have the small letters together. You have to make sure the small letters are not together. Again, you put this, when you have a capital letter, basically it suppresses the effect of the small letter. You should not give the answer like this. In this case, the person would be diseased. But over here, do you think the person is diseased? No. Okay, in the pedigree chart, autosomal recessive disorder is going, but this person does not have it. That means this should be the thing. Sex-linked dominant disorder. Now, sex-linked dominant disorder means, basically suppose it should be on X chromosome. It should be on X chromosome, and basically this X chromosome which is there, it should be affected with the gene. So if you make it generally like this, that also basically works. Okay, it also basically works. Suppose, okay, wait just a second. X-linked dominant disorder, you can either show it like this, and you can mention that A and A. This is the dominant allele and this is the recessive allele. In that case, this person does not have, this person has small allele that does not have the dominant allele, which is why this person is safe. That is exactly being mentioned over there. Now, X-linked recessive disorder. X-linked recessive means, again, as I said, it should not, over here both of them are disease. So you should show the disease which is X-linked recessive. So here, a person who is still having this would be having the disease because it is recessive disorder. Again, a person like this will be also having the disorder because both the recessive alleles are coming together. So this is kind of tricky, but this is how you need to frame the answer. So this does not depend upon this, you also have to look on this particular diagram. Cool? Cool. Now next thing, question number 44. Justify the below given statements. RNA polymerase cannot transcribe a gene by itself. Can it? No. It needs the help of a factor which is known as sigma factor. Sigma factor helps for attachment of the RNA polymerase into the promoter region, and that's where the transcription officially begins. And then you need rho factor for terminating the process of transcription as well. That is being asked over here. Now transcription and translation are separated by site and not coupled in eukaryotes. Yes, you have to explain that. So you have to say in prokaryotes, since there is no nucleus and cytoplasm, everything happens in one region. But in eukaryotes, since you have a nucleus and everything and the cytoplasm separate, so transcription happens inside, okay, DNA to RNA, mRNA formation, and this mRNA moves outside for the process of translation. Now, which RNA polymerase enzyme is involved in transcription of srRNA? srRNA. So it is capital S actually. Okay, and the answer for this 5S, okay, so it will be 5S. 5S rRNA, it is actually RNA polymerase 3. So along with tRNA, RNA polymerase 3 also is responsible for 5S rRNA, the only rRNA. Usually rRNA is being transcribed with RNA polymerase 1 itself, but this is an exception. So first thing, RNA polymerase cannot transcribe a gene by itself because it cannot recognize the promoter region of DNA by itself. It requires auxiliary proteins such as sigma factor to bind correctly to the promoter and start the transcription. Okay, now transcription and translation are separated by site and not coupled in eukaryotes. Transcription occurs in the nucleus and translation occurs in the cytoplasm. Also, the primary RNA undergoes processing, that is capping, tailing, splicing, before leaving the nucleus. Because they occur in different cellular compartments and RNA must be processed first, transcription and translation are not coupled in the case of eukaryotes. They have to be done in the separate regions. Now, RNA polymerase 3 is involved in transcription of 5S rRNA. This is a simple question. Now, there are some questions for visually challenged students. So, they are replaced. For example, the 11th question which is there, this is something else for the visually challenged students. So, the chemical nature of primer used in polymerase chain reaction. So, when you make the primer out there in PCR, polymerase chain reaction, usually primers are made up of RNA. But over here, the primers are basically made up of deoxyribonucleotides. Not the ribonucleotides, but deoxyribonucleotides in the case of polymerase chain reaction. Always remember that. Okay. Now, enlist the steps of recombinant DNA technology. So, you have to talk about that also. This is replacement of 32nd question. So, what happens in recombinant DNA technology? First of all, you are going to isolate the DNA. So, the DNA might be clubbed with proteins, RNA, everything. So, depending on whatever organism it is, first of all, suppose it's a plant cell. So, you cannot directly get the DNA. Initially, you have to break the cell wall. The cell wall is made up of cellulose. So, you have to take it with cellulase. Okay, and then basically you have RNA, protein. So, RNA is treated with RNase, proteins are treated with protease, and last thing that you get is what? The DNA. The DNA. Okay, so you have to put chilled ethanol, the DNA will be separated. That's how you do isolation of DNA. Now, once you gotten the DNA, you're going to cut it with the help of restriction enzymes. Okay, you're going to cut it with restriction enzymes. Then you're going to separate the fragments with the help of gel electrophoresis. After that, you're going to pick the unit and put it inside the vector which is going to transfer. Suppose I want to transfer it to B organism. I cannot directly put it in the B organism. I need a vector for that. Suppose it's an animal, then I'll use retroviruses. Suppose it's plants, I'll use agrobacterium tumefactions. Okay, so now this particular vector will be having my gene of interest. So my first thing is insertion of the DNA into the vector. So that has to be done. After inserting, okay, I think I have missed the other one. I'll give it the PDF in the PDF version, don't worry. After inserting my DHL, what will happen? What will happen? This particular vector is going to transport it to the organism where we need. And basically in that organism it is going to divide, replication would be happening, multiple copies are made, and then downstream processing and everything is being done. Whatever thing we need is being gotten from the bacterial cell or any other cell where we have inserted this particular thing. Okay. So these are the steps of recombinant DNA technology, all of which I have actually explained really well in the Brahmastra series. You can go watch the question. I have done it chapter wise. It is going to be super helpful for you guys. Okay, now 38th question, I think that marks the end of it. Name the cells involved in humoral and cell-mediated immunity. So humoral means basically you're talking about antibodies. So in our humor, in our blood, what do we have for protection? We have antibodies. So cells involved in humoral, who releases the antibodies? It is the B lymphocytes. Now cells involved in cell-mediated immunity means basically, cell-mediated immunity is basically T cell mediated. As I said, T cells tells the other cell you have to produce this, you have to secrete this, all of that. So it is T lymphocytes. Okay, both of them are a part of what? Part of what immunity? Innate and acquired. Acquired immunity. It is a part of acquired immunity. Okay, I have one more question. Fine. Mention the salient features of Down syndrome. So what do you find? Mental retardation, short stature would be there, broad palm with prominent palm crease will be there, partially open mouth will be there, physical, psychomotor and mental retardation or development would be found. Now what is the karyotype of Down syndrome? what is the cause of Down syndrome? That is the first question. I'll answer that first. So initially we have 21st chromosome. If we see, we get one copy from one parent, another copy from another parent. But over here you have one more extra copy. Now you call this trisomy of 21st chromosome. Trisomy of 21st chromosome. Now you can make the karyotype. Usually we have how many autosomes, my dear children? Normal chromosomes, which are not sex chromosomes. We have 22 of them. Our 23rd pair, which is there, 23rd is the sex chromosome. Okay, 23rd which is there, that is sex chromosome. So out of 46, if you are talking, 44 of them, 22 into 2, 44, right? 44 of them are autosomes, and 45th and 46 are the sex chromosomes. So over here, since we have one extra one, so it will be 45, not 44 but 45, because autosome is extra. Now if you talk about the sex chromosome, it can be 45 XY. In that case, it is what? 45 autosomes plus XY. That means it is a male who is having Down syndrome, XY. Now it can be 45A plus XX. That means it's a female who is having Down syndrome. So you have to write like that. Or you can write it like this also, but that does not make sense. It is more correct for you to write this. So 45A plus XY, it is a male. And 45A plus XX, it is going to be a female. Cool? Yes. And with that, we complete the model question paper 1 of biology. I hope you have understood certain things. I tried to explain all of it. I have given even more detailed explanation in the Brahmastra. Go watch it. I didn't go in depth because it might take too much time and you might not have that capacity to stay hold of longer duration videos. But yes, so I just want to say wishing you all the best, do well, and make sure you practice all the model question papers, and please retry this question paper as well. Wishing you all the best. Take care. Bye-bye.